Hi everybody. So in this lecture, I'm just covering section 2 of chapter 18, which discusses the electron transport chain. In the next lecture, we'll cover sections 3 and 4, which discuss oxidative phosphorylation, so making ATP through oxidative phosphorylation versus substrate level phosphorylation. And then chapter 4 discusses the regulation of that process. Okay, because every metabolic pathway we're going to talk about how is it regulated. All right. So section 1 is a great review on the mitochondria. So if you can't remember much from biology, maybe go back to section 1 and read over that so you can familiarize yourself with the mitochondria and the role that the membranes of the mitochondria play. All right. So here are our learning objectives for this chapter. Just got a few different ones here for you to look over. I'll pause for a second so that you can pause and print. Alrighty, continuing on. Alright, so the first thing to know, obviously, is that the electron transport chain is exergonic. If it wasn't exergonic, we wouldn't be making ATP, would we? So, as you can see, electron carriers, such as NADH and FADH2, carry electrons to our end electron acceptor O2. So this is obviously a part or near the ending of aerobic respiration. So associated with the inner mitochondrial membrane, all of these electron transport proteins are embedded in this membrane. And we see that there are a sequence of carriers and that these carriers reflect the relative reduction potentials so that the overall process is therefore exergonic. So as we see, an oxidized substrate's affinity for electrons increase with its standard reduction potential. So if we go and look at these half potential reactions that we kind of looked at back in the chapter on energy, or the chapter on the introduction of metabolism, we can see if we look at the half reaction of NADH plus H plus, which is the reverse of the reduction of NAD plus. If we reverse that for the oxidation, we see that we get a plus 0.32 reduction potential. The reduction of oxygen in order to make H2O gives us a reduction potential of 0.816. If we plug this, or if we total that change in energy, we see that we get plus 1.136 volts. If we plug that delta E into our Nernst equation for the Gibbs free energy under standard conditions, we see that we get an overall delta G of negative 219 kilojoules per mole for every molecule of NADH we oxidize in order to reduce oxygen into water. So that's a lot of energy. So that's 219 kilojoules per mole of energy released. Again, we see, so just to recap, the transfer of electrons from NADH to O2 can release up to 219 kilojoules per mole of energy. Now remember, this is under standard Gibbs free energy. If we require 30.5 kilojoules per mole of energy to make one molecule of ATP, take a moment and calculate how many ATP can we theoretically make. Alright, so I'll let you come into class and tell me about that. Now, on average, in the cell, we actually only make 2.5 molecules of ATP per molecule of NADH. So if that is true, what is the actual oxidative efficiency under standard conditions of the oxidation of NADH, or of the reduction, or no, the oxidation, I can't even get it right sometimes. So what is the oxidative efficiency of the oxidation of NADH to O2 in order to make ATP. Again, that's something that y'all can calculate and then come talk to me in class, but I will give you a hint if you look at section 2A. Okay, so take a look at that section and you might be able to find a way to approach this problem. Alright, so again, just to reiterate, electron carriers operate in sequence. Again, we want to increase our reduction potential as we go down the sequence. The electron transport chain is a set of protein complexes containing redox centers with progressively greater affinity for electrons, starting with complex 1 and complex 2. So complex 1 transfers electrons from NADH, where complex 2 actually transfers electrons from FADH2 through succinate to fumarate, and we'll talk about that if you recognize that. Both of these complexes transfer their electrons to coenzyme Q. Coenzyme Q then goes to complex 3 and transfers its electrons through its different redox centers to go eventually to cytochrome C, and then cytochrome C swims its way over to complex 4, which then the electrons pass through these cytochrome centers eventually to oxygen and making water. All in all, throughout this process, we're also pumping protons from the matrix into the intermembrane space of the mitochondria. Okay? And we'll talk about why that's very important when we talk about oxidative phosphorylation in section 3. Okay? All right. So another way to look at the electron transport chain is in this figure here in your textbook, figure 18.7. And then we can also look at different things that inhibit. So instead of normal regulatory inhibitors, what we're talking about here is actually toxins or poisons that can inhibit the electron transport chain. We'll talk about other ways that we can regulate this in section 4 in the later lecture, but a lot of these toxins or poisons were used in order to elucidate the different functioning complexes of the electron transport chain, and that's why it's mentioned in the textbook. So both rotonin, which is a plant toxin, and amytal, which is a barbiturate, both block transport in complex 1. Okay. Antimycin A, which is an antibiotic, blocks complex 3. And cyanide blocks complex 4. The reason cyanide is so lethal is because if we block any of these complexes, we can eventually come in other complexes. So if we block complex 1, we can still come in at succinate with FADH2 and continue the cascade down. However, if we block complex 4, this is sort of our end game in the electron transport chain, and thus we will completely stop any ATP development through the electron transport chain, definitely causing damage to the cell because we're not going to make enough ATP from just glycolysis alone. One thing I did want to note, if y'all were following Brittany Menard, if you know her Death with Dignity, the lethal medication that she took was an overdose of barbiturates, and obviously one of the side effects of barbiturates is blocking electron transport, thus reducing ATP, thus arresting the cells in that state. Alright, so complex 1, let's take a deeper look into complex 1. So this is that first complex in the electron transport chain. It's also called NADH Coenzyme Q Oxido Reductase. So obviously from its name, you can elucidate that complex 1 passes electrons from NADH to coenzyme Q. It does this through its multiple coenzymes. Flavin mononucleotide, which is the first center to accept electrons. It can accept either 1 or 2 electrons. FMN then passes its electrons over to iron-sulfur clusters. These can only undergo 1 electron redox, so you have to pass the electron one at a time from FMN to FES. The FES clusters then transfer their electrons over to coenzyme Q. In addition, during this, 4 protons are pumped out of the matrix into the intermembrane space, and we'll talk about why that's important, remember, in oxidative phosphorylation. Complex 2, which is succinate coenzyme Q oxidoreductase. Do you recognize the metabolite succinate? Hopefully you do recognize it from the citric acid cycle. Complex 2 contains the TCA enzyme succinate dehydrogenase. It's actually covalently bound. In that reaction, electrons pass from succinate to a covalently bound molecule of FADH2, or of FADH in order to make FADH2. Those electrons then pass through iron-sulfur clusters and cytochrome B560, and then eventually to CoQ. So CoQ is this sort of overall electron acceptor from either NADH or FADH2, and which is really good because this enzyme is lipid soluble and is able to freely diffuse through the membrane of the mitochondria, and allows it to go between complexes in order to transport the electrons from either complex 1 or complex 2 to complex 3. Here we can see an idea of that cascading of electrons. So succinate transfers its electrons from FAD to make FADH2, and then the electrons will then cascade down the iron clusters, eventually to heme3, which is this cytochromes BH, and then eventually, that's the wrong complex. Anyway, cascades down to that heme3, which is the cytochrome B560, and then to coenzyme Q. All right. Complex 3 is also called coenzyme Q cytochrome C oxidoreductase. So here electrons pass from CoQ to cytochrome C. So we have CoQ gives its protons or gives its electrons to complex 3 through all these different cytochromes, and then eventually to cytochrome C, which is again a mobile protein on the membrane that can go between complex 3 and complex 4. Complex 3 contains 2 B-type cytochromes, 1 cytochrome C, and 1 iron sulfide cluster. The reduced form of CoQ, CoQH2, is a 2-electron carrier, remember? So it reduces 2 molecules of cytochrome C, a 1-electron carrier, in what is called the Q cycle. This cycle allows complex 3 to pump protons from the matrix into the intermembrane space. So now we have another site of proton pumping. As you see, we get another 4 protons from complex 3. As you see, for each electron in the pair, we get 2 protons for a total of 4. All right. Complex 4 is also called cytochrome C oxidase. This catalyzes the 1-electron oxidation of 4 consecutive reduced cytochrome C molecules. So our cytochrome C, remember, only carries 1 electron from complex 3 to complex 4. So complex 4 requires a total of 4 cytochrome C molecules. So it needs 4 electrons in order to reduce 1 molecule of O2 into water. this complex contains 4 redox centers: cytochrome A, A3, copper B, and a copper A clustered center. Cytochrome C oxidase has 2 proton translocating channels. First, protons from the matrix are used to form water upon the oxidation of O2, and the 4-electron reduction reaction translocates 4 additional protons out of the matrix into the inner space. Therefore, for each molecule of oxygen that we oxidize, the matrix loses, for each molecule of oxygen that we reduce, the matrix loses a total of 8 positive charges. And again, remember, we'll see why this matters in the next section. So if we look at our overall reaction for complex 4, we have 8 protons from the matrix, 1 molecule of water. We require 4 cytochrome, reduced cytochrome Cs. So we say that our iron is reduced from 3+ to 2+. So we have 4 electrons total. This gives us back 4 oxidized cytochrome Cs. So see, our iron is now a 3+ because we've lost an electron on that iron center. We get 2 molecules of water total, and then 4 protons are pumped from the matrix into the intermembrane space, starting to create our proton gradient that we'll see that's important in oxidative phosphorylation. So if you think our intermembrane space is starting to get really positive and filled with a lot of protons. Alright, so basically that's all we needed to cover in this section. There is a lot of detail in this chapter. As long as you can discuss the learning objectives that are at the beginning of this lecture, you should be fine. And I will look forward to talking with you about the questions that I asked in this lecture, as well as the activity that we're going to go over in class. Okay, so I will see y'all then. And